DNA replication starts out as one double helix template strand with parental 3 and parental 5 strands and is eventually unwinded by helicase. This process is done with the use of DNA polymerase III. After helicase unzips the DNA, single-strand binding proteins are added to the split DNA. They will hold the two strands apart from each other to make sure that the strands do not reattach, which would then result in not being able to replicate. This is called continuous synthesis. When RNA primer is added, the DNA polymerase now has a binding site where it can attach to the DNA and add nucleotides to the synthesized strand. This process is catalyzed by the primase, which initiates the synthesis of DNA. Once the primer is added, the DNA polymerase can fold the helicase complex, and as the DNA gets unzipped, the polymerase follows behind, adding sequential nucleotides. The DNA polymerase I replaces RNA primers with DNA. For the lagging strand, it is added in fragments so that as the DNA gets unzipped, the template strand is moving right to left, and the DNA polymerase cannot add new bases in the same direction, so it is going to have to travel from left to right to add new nucleotides. This is called discontinuous synthesis. As the DNA helicase is unzipped, the RNA primer is added so a DNA helicase can come in and synthesize a small segment of DNA up until it bumps into another one of the Osaki fragments. The ligase links these Osaki fragments, and DNA polymerase will add new strands until it bumps into the RNA primer, in which case it will detach and stop, and later reattach to other pieces.